Welcome to the new video on the history of bioinformatics. The history of bioinformatics is quite complex as it shares the events with two different branches of science. One is biological science, the other one is computational science. In this video, I will only mention the major milestones of bioinformatics. Bioinformatics has become so robust because of the revolution of technologies in both these disciplines. This is fueled by the demand of products both in biotechnology and medical sciences. In addition to it, the internet technology has augmented the rapid growth of bioinformatics. The word bioinformatics was first formally used by Paulien, Hogeweg and Ben Hesper in 1970 to refer to the study of information processes in biotic systems. Bioinformatics involves the analysis of biological data, particularly DNA, RNA and protein sequences. The field of bioinformatics experienced explosive growth in the mid-1990s, driven largely by the Human Genome Projects and by rapid advancements in DNA sequencing technologies. Analyzing biological data to produce meaningful information involves writing and running software programs that use algorithms from graph theory, artificial intelligence, soft computing, data mining, image processing and computer simulation. The algorithms in turn depend on theoretical foundations such as discrete mathematics, control theory, system theory, information theory and statistics. Computers and specialized softwares have become an essential part of biologists' toolkit, either for routine DNA or protein sequence analysis or to pass meaningful information in massive gigabyte-sized biological data sets. Virtually all modern research projects in biology require to some extent the use of computers and softwares. A proper foundation for bioinformatics was laid by the accumulation of information and technologies in two branches of science. One is biology, the other one is information technology. The genetic basis of life was validated by four independent experiments. One was the experiment by Hershey and Chase in 1952. They validated that the DNA is the molecule which stores the genetic information. Later in 1953, Watson and Crick proposed the DNA double helix model, which explained the chemical nature of DNA. It's after 13 years from the time when DNA double helix model was proposed, that is in 1966, the genetic code was deciphered by Halley, Nirenberg and Khorana. And it is only after 25 years after the DNA double helix model was proposed, it was in 1978, the methods of DNA sequencing became available for everyone. Margaret Dayhoff pioneered the application of computational methods to the field of biochemistry. Her contribution to this field is so important that Lippmann, the former director of NCBI, called her the mother and father of bioinformatics. Margaret Dayhoff is known as the first bioinformatician. From 1958 to 1962, along with Reddy, she developed the very first software for bioinformatics called COM-Protein. It is designed to determine the protein primary structure using Edman peptide sequencing data. Margaret Dayhoff also developed the very first database in bioinformatics called Atlas of Protein Sequence. It used one-letter code for the first time in 1965 and developed the Atlas of Protein Sequence and Structure. The first edition of the Atlas combined 68 protein sequences, most of which were interspecific variants of a handful of proteins. Zucker and Paulien showed us how to use information technology or computers to understand the ancestry of proteins, ancestral sequences, everything, and it led to the formation of a new branch called evolutionary biology powered by bioinformatics. They studied the evolution of hemoglobin by using information technology and bioinformatics. At present, there are thousands of software programs and tools and techniques and algorithms that are used in bioinformatics. The first two very important algorithms that were developed specifically for bioinformatics were Needleman and Wunsch algorithm, and at the same time there's one more algorithm called Smith and Waterman algorithm. These two algorithms are used for local and global alignment, and these are the two important methods of sequence similarity search that is done in bioinformatics. Flow of information in living systems occurs from DNA to RNA to protein. This concept was hypothesized by Crick and it was called central dogma, and based on this all the other further studies were made, and this also is an important milestone in genetics and also in biology. DNA sequencing is one of the major activities that occurs in bioinformatics, and the first method of DNA sequencing was developed by Maxim Gilbert in 1976, and this has led to a revolution in biotechnology. Many more methods of DNA sequencing are made available. In 1977, Sanger developed one more method which is popularly known as Sanger's method of DNA sequencing, with the help of which they sequenced the bacteriophage CX174. This is the first ever organism to be completely sequenced. Staden package is one of the first sequence analysis software to include additional characters to record base calling uncertainties in a sequence read. This was useful and this augmented the method of DNA sequencing, and it was quite useful in the improvement of the methods used in DNA sequencing. The second milestone in manipulating DNA was the polymerase chain reaction, popularly known as PCR, which allows the DNA to be amplified without cloning procedures. The invention of PCR was done by Kary Muller. He notably used the thermolabile Taq polymerase, which is working at higher temperatures, which is quite essential for PCR reaction. The next step in DNA technologies is the DNA manipulation or genetic manipulation. One of the first technique to manipulate DNA was developed by Jackson, Simons and Berg in 1972. They used restriction endonucleases and DNA ligases to cut the DNA and to rejoin DNA. This led to a revolution in biotechnology, and that is genetic manipulation or DNA manipulation, with the help of which you can modify the organism, you can modify the genetic information, which is a very common thing nowadays. Further growth and development of bioinformatics was augmented by the accessibility to mini computers and specialized softwares. The first mini computer was manufactured in 1965, but the first wave of ready-to-use microcomputers hit the consumer market only in 1977. These mini-computers had a built-in basic interpreter, which was an easy language for non-programmers, which helped the improvement in bioinformatics. Microcomputer software called GCG software suite was developed in 1984. This was the first software collection developed for sequence analysis. Free software movement was started in 1985 by Richard Stallman. Because of this free software foundation, users had the freedom to run, copy, distribute, study, change, and improve the software. In the late 1980s, the major biological databases NCBI, EMBL, DDBJ, and also INSDC were established. Late 1980s were very crucial for bioinformatics, and in late 1980s the major biological databases like NCBI, EMBL, DDBJ were established. The collaboration of all these major databases formed a union called INSDC, the International Nucleotide Sequence Database Collaboration. Also, during this period, the dedicated journals for bioinformatics were also established. Rapid growth of bioinformatics was also helped by the desktop computers and new programming languages which occurred in 1980s and early 1990s. First programming language that was used for bioinformatics was Perl, practical extraction and reporting language. At present, all the three major programming languages like BioPerl, BioJava, and BioPython are being used extensively in bioinformatics. The most important period for bioinformatics is a decade from 1990 to 2000. This period is popularly known as genomics, structural bioinformatics, and information superhighway. In this period, from 1990 to 2000, three things occurred. Number one, the establishment of internet, dawn of genomics era, and windfall of bioinformatics tools. BLAST, Basic Local Alignment Search Tool, is the most sought after search tools in bioinformatics that was also developed during this period. During this decade, we also saw a revolution in DNA sequencing technologies. Many technologies, many methods were developed for sequencing the DNA. Even the cost of sequencing was also considerably reduced during this period. During this decade, bioinformatics almost became completely online. In 1992, Gene Bank database under NCBI was established. The next decade from 2000 to 2010 is known as high throughput bioinformatics. It's because of the improvement in DNA sequencing methods and also improvement in NGS, that is next generation sequencing. NGS allows sequencing thousands to millions of DNA molecules in a single machine run, which revolutionized sequencing technology and also bioinformatics. Because of the revolution in bioinformatics, the amount of sequence data that is made available in public databases is humongous and hilarious. So thus, bioinformatics has grown enormously because of the development of technologies both in computer science, information technology, and also biological sciences. Two major breakthroughs or achievements of bioinformatics that are worth mentioning are: number one is the completion of Human Genome Project, and the number two is development of CRISPR, the gene editing tool or gene editing technology. With the help of these two achievements, we can solve, we can diagnose, we can treat, we can cure most of the human diseases, most of the problems of mankind. The period from 2010 to 2023 belongs to bioinformatics. Trillions of sequences were analyzed, thousands of genomes were sequenced, hundreds of diseases were clearly understood, hundreds of drugs were designed and produced, several genetically modified organisms and biological products were produced. All this is because of tremendous advancements in the technology. Undoubtedly, the future belongs to bioinformatics. There will be millions of job opportunities, startup options, commercial avenues. At present, the focus of bioinformatics is on medical, biotechnological and environmental engineering. The period from 2010 to 2023 has been tremendous for bioinformatics. Thank you very much for watching my videos. Please like, share and subscribe. We'll meet again in the next video with some other topic. Thank you very much.